All right, let's dive right in. Today we are cracking open the secret life of pathogens. We're going to explore how the very same microscopic bug can be totally harmless in one place, but a really serious threat somewhere else. You know, it's a story that's written in DNA, and we've got the decoder ring.
So let's start with the central mystery, right? How can one type of bacteria act so completely differently depending on where it is? I mean, think about it. A human, a cow, a puddle of water, it's a question that has massive consequences for our health, because understanding this is really the key to fighting infectious disease.
And the answer, it turns out, is a full-blown genetic detective story. See, bacteria aren't static. They are constantly editing their own instruction manuals, their own DNA. They're adding new tools, tossing out old ones, all to become absolute masters of their domain, whether that domain is inside your lungs or just a patch of soil.
So how in the world do scientists actually read these constantly changing blueprints? Well, the main tool in their detective kit is something called comparative genomics. It's kind of like comparing different editions of a book to see where the author, in this case, the bacteria, made edits to adapt the story for a brand new audience. Now, to solve a mystery this big, you need a mountain of evidence. And in this case, the evidence was this: 4,366 high-quality bacterial genomes. That is an absolutely incredible amount of data, giving us this panoramic view of the microbial world.
The whole investigative process itself is actually pretty simple and elegant. First, you gather all your genetic evidence. You know those thousands of genomes? Then you sort them into 3 different groups based on where they were found: inside a human, inside an animal, or just out in the environment. And finally, you run this massive comparison to spot the differences, to find the specific genes that define each lifestyle.
So what did the detectives actually find after sifting through all that DNA? Well, they discovered that bacteria in each of these 3 places, human, animal, and environment, are basically operating from completely different strategic playbooks. And at the very heart of these playbooks are 2 fundamental moves, and they're summed up perfectly in this quote from the source material. Bacteria adapt by either getting new genes to gain new abilities, or by losing genes to become more streamlined and efficient. It's this constant balancing act between being a jack of all trades and a master of one.
And when we put these strategies side by side, the differences are just striking. Bacteria living inside us humans, they're master gene traders. They are constantly acquiring new genetic tools, kind of like a spy picking up new gadgets to help them dodge our immune systems and stick to our cells. Now, in contrast, bacteria out in the environment are more like survivalists. They're generalists who keep this broad, stable set of tools to handle whatever unpredictable stuff comes their way. But, and this is what's really fascinating, some bacteria, to become true human specialists, do something that sounds totally backwards. They actually lose genes. They streamline their genetic code, throwing out tools they don't need anymore to become hyper-efficient inside the stable, predictable environment of our bodies.
This specialization also extends to their weapons, or what scientists call virulence factors. As you can see here, human-associated bacteria, that top row, they prioritize tools for sticking to us and hiding from our immune system. They're built for stealth. But take a look at that middle row for the animal niche. It acts as a huge reservoir for some of the most dangerous offensive weapons, like exotoxins. This is a critical point. The tools that can make us really sick are often being stored and shared in the animal world, which just shows you how connected all these environments are.
This research also shines a huge light on one of the biggest challenges in modern medicine: antibiotic resistance. And this chart just makes the story crystal clear. I mean, look at that first bar on the left. Bacteria that come from humans carry the highest load of resistance genes by far. This is evolution happening in real time, folks, driven by our use of antibiotics. And notice the second bar. Animal-associated bacteria aren't far behind, which just reinforces their role as this critical place where these resistance tools can be stored and shared.
So beyond these big, broad patterns, the genomic detectives found a specific, repeating clue, a potential smoking gun that pointed to a key mechanism for how bacteria adapt to humans.
Sifting through all that data, they noticed one particular signature gene kept popping up over and over again. But here's the thing. It was almost exclusively in the bacteria that had made a home inside humans. That's not a coincidence. That is a powerful, powerful clue.
The clue was a single gene, Hype B. And this gene is a total game changer. It's not just involved in basic energy metabolism, it's thought to be a key part of the bacteria's invisibility cloak, helping it hide from our immune system. This makes it a potential master key,
something that allows a whole range of bacteria to unlock the human body as a new, resource-rich environment to call home. Okay, so this is so much more than just an academic exercise in decoding genes. This is actionable intelligence. This is information that has profound implications for all of our health.
Because what this research truly proves, down to the molecular level, is the One Health concept. You just can't separate the health of humans from the health of animals and the environment we all share. Pathogens and their genetic toolkits are constantly flowing between these three worlds, and this study basically gives us a map of those pathways.
And with that map, we can start to take action. We can get better at predicting how pathogens might jump from animals to humans. We can develop smarter antibiotic strategies by understanding the specific resistance tools of bacteria in different places. We can even design targeted treatments that go after a pathogen-specific playbook. I mean, imagine a drug that specifically targets that high PSB gene, disarming bacteria that are specialized for living in us. This is the future of fighting infectious disease.
So by decoding these genetic playbooks, we get this unprecedented look into the strategies that pathogens use to survive and to thrive. But bacteria never stop evolving. They're constantly writing the next chapter. This research gives us the tools to read their current strategy, which is really the only way we can hope to anticipate their next move.